Okay, chapter 11, neuromuscular system and exercise. All right, objectives today are identify the major structural components of the central nervous system, control human movement, diagram the anterior motor neuron, discuss its role in human movement, draw and label basic components of a reflex arc, define motor unit, neuromuscular junction, autonomic nervous system, excitatory postsynaptic potential, and inhibitory postsynaptic potential. Explain factors associated with neuromuscular fatigue. Describe the function of muscle spindles and Golgi tendon organs. Draw and label skeletal muscle fibers, ultrastructural components. Describe the sequence of chemical and mechanical events during skeletal muscle contraction and relaxation. Contrast slow twitch, fast twitch, including subdivisions, muscle fiber characteristics, outline muscle fiber type distribution patterns among diverse groups of elite athletes, explain how exercise training modifies muscle fibers and muscle fiber types. Muscle fibers and fiber types. Nervous system, you have the central nervous system, brain, spinal cord, and then peripheral nervous system, PNS, which is cranial nerves, spinal nerves. Okay. So the nervous system, you have the brain, spinal cord, vertebral column, spinal nerves. Peripheral nervous system has the so the peripheral nervous system includes the cranial nerves 3 to 12, and the spinal nerves, and that deals with the efferent division or the motor neurons that conduct impulses from the CNS to the effectors. And that deals with the autonomic nervous system and the somatic. Autonomic is like the involuntary control, impulses, cardiac muscle, heartbeat, rhythm, smooth muscles, glands, that's autonomic split into parasympathetic, sympathetic types. And then somatic nervous system is voluntary, conducts impulses from central nervous system, the skeletal muscles. And then you have the central nervous system, including the brain, retina, spinal cord, integrative control center, afferent, sensory, somatic and visceral neurons, conducts impulses from the receptors to the central nervous system. So we'll go over all this. Central nervous system, the brain, you have 6 major divisions. You have the medulla oblongata, pons, midbrain, cerebellum, diencephalon, telencephalon. You have the longitudinal fissure, right and left hemispheres, cerebral cortex, gray matter, occipital, parietal, temporal, and frontal areas. And this is on page 322, and it describes all the portions of the brain. Okay. In regards to this, vestibular area, by the way, is spatial orientation or balance, if that term is not familiar to you. CNS or spinal cord encased by 33 separate vertebrae. 33 vertebrae separated by intervertebral discs. The ventral horn, dorsal horn, which includes the inner neurons, sensory neurons, motor neurons. Pyramidal tract is transmit impulses down through the spinal cord. That basically controls voluntary reflexes. Okay, and then you have the extrapyramidal tract, which the neurons originate in the primary upper motor cortex of the brain and connect all spinal levels. This is involuntary movements like posture, continual background level of neuromuscular tone. CNS, spinal cord, you have cerebrum, cerebellum. You have the spinal cord ventral view. So you have this individual going up for a pole vault. What happens is there's a signal that's relayed to the spinal cord through an afferent source. It goes into the spinal cord, comes back to the muscle to then produce a movement. It's basically as simple as that. And then you have lumbar vertebrae and then cervical vertebrae, which have different parts labeled. And this is on in your book too as well on page 323. All right. Again, to break it down a little bit with the spinal column, you have the human spinal cord showing peripheral nerves, and then ventral view of the ventral view there. Of the spinal cord section to illustrate dorsal and ventral root neural pathway and nerve nerve impulse direction. Okay, which we talked about. It comes from seeing that there needs to be recruitment of muscle fibers. There's a signal sent to the spinal cord, and then it reverts back. So it goes from an efferent pathway to an afferent, back to the spinal cord to an efferent, to then recruit these muscle fibers to create a movement. Brain neurotransmitters, so you have chemical messengers diffuse across the synapse, combined with targeted receptor molecule in the postsynaptic membrane, facilitate depolarization, hyperpolarization. So there's three let me go back here for a minute because I want to explain something before. Okay, sensory neurons and motor neurons. Should say motor, by the way. So you have motor efferent neurons conduct impulses outward from the brain to the spinal cord. Okay. They exit the cord through the ventral root to supply extrafacial and interfacial skeletal muscle fibers. Sensory afferent neurons enter the spinal cord via the dorsal root and carry impulses from sensory receptors toward the CNS. So what we're saying is that you have these sensory neurons. When you go back to here, sensory neurons travel towards the CNS, and then the CNS elicits a response for the motor efferent neurons to then elicit the response, which is the movement of the individual moving the muscle, basically, to create a reflex towards whatever the individual has to do, like the person that's trying to use the pole vault. Okay, here you have monoamines, which, okay, these are amino acids. Okay, they include epinephrine, which is adrenaline, norepinephrine, serotonin, histamine, dopamine. Okay, three important neurotransmitters that create these to depolarize or hyperpolarize the environment. Then you have neuropeptides, arginine, vasopressin, angiotensin 2, enkephalins, and endorphins. So the endorphins you see there's well, let me back up. Monoamines. Okay, so you have serotonin, you have histamine, dopamine. The thing about serotonin is an increase of that elicits a decrease in the stress response. You have dopamine, which increase of that causes focus. Norepinephrine causes focus. Epinephrine is adrenaline. So a whole bunch of things that take place here in these neurotransmitters that then can either depolarize or hyperpolarize different organs. Okay. You have arginine, vasopressin, angiotensin 2, enkephalins, endorphins. So endorphins, if you've heard of it before, it release of endogenous opioid transmitters with physical activity contributes to exercise high. That's an endorphin, beta endorphin. And nitric oxide actually is a substance that causes a vasodilator effect. Okay, where it causes your vessels to widen, and it's so all these things have effects that are caused by the signaling of, let's say, some type of, okay, you see a situation where you have to move quickly, and because of that, then there's this cascade effect of neurotransmitters that are released endogenously from the brain. Okay, you have the peripheral nervous system. Okay, this contains spinal nerve, 31 pairs of spinal nerves, 8 cervical, 12 thoracic, 5 lumbar, 5 sacral, and 1 coccygeal. 12 pairs of cranial nerves include afferent nerves that relay sensory information from muscles, joints, skin, and bones toward the brain. If it's going toward the brain, it's afferent. Okay. Okay, so that's like, okay, I see that I have to move my hand to grab something. So there's my hand, basically there's information, okay, from the muscles that goes up to the brain. Then efferent nerves transmit that information away from the brain to the glands and muscles to then cause that movement. Okay, you have somatic and automatic nervous systems. Somatic nervous system innervates skeletal muscle. Somatic efferent nerve firing excites muscle activation. Okay, and so you have autonomic nervous system, which innervates smooth muscles in the intestines, sweat, and salivary glands, myocardium, seminecine glands. Functions as a unit to maintain consistency in the internal environment. And here you have the so the one thing about this with the somatic, this is a voluntary nervous system. Okay. Somatic efferent nerve firing excites muscle activation. Autonomic nerve, okay, either excites or inhibits. So you have the autonomic nervous system that consists of the sympathetic and parasympathetic nervous system. Okay, you have the comparisons are basically the sympathetic nerve fibers excite, parasympathetic inhibit. Sympathetic deals with, you have your adrenaline. Parasympathetic deals with acetylcholine. Okay. Or should I say epinephrine? So you have sympathetic deals with epinephrine, and then acetylcholine is what's used to elicit this parasympathetic response. And that's what we see here. So if you have comparisons here, you have effective sympathetic, parasympathetic activation on organs. You have end organ, skeletal muscle. What does sympathetic effects do? Increases blood flow. Parasympathetic decreases blood flow. Okay, ventilation. Sympathetic effects increase ventilation. Decrease happens with the parasympathetic effects. Okay, you have the eyes are dilated with sympathetic, constricts with the parasympathetic. Blood pressure increases with sympathetic, decreases with parasympathetic. And then you have airways, increased diameter with sympathetic and decreased diameter with parasympathetic effects. And this really here, I like, this tells you up here a bit what takes place. So you have the parasympathetic division. Look at that. It's coming from, a lot of it comes from up to, in the brainstem. Okay, you have in the cranial area, that's where a lot of the directions coming from parasympathetic. Okay, that's the easiest way to put it. And the sympathetic division is, you have the thoracic, okay, causing a lot of these directing these effects. Sympathetic nervous system. Okay, so let's talk about it again. So supply the heart, smooth muscle, sweat glands, viscera. Neurons exit spinal cord, enter a series of ganglia near the cord's sympathetic chain. Nerves terminate relatively far from the target organ. In adrenergic fiber endings that release norepinephrine. Excitation occurs during fight or flight situations that require whole body arousal for emergencies. This accelerates breathing and heart rate. Pupils dilate and blood flows from the skin to deeper tissues. Parasympathetic nervous system, neurons exit the brainstem, sacral segments the spinal cord to supplies thorax, abdomen, pelvic regions, release acetylcholine, postganglionic parasympathetic nerve fibers produce effects opposite to sympathetic fibers, which we talked about that. Parasympathetic nervous system. So then you have this here you have the in the doctor's office you have a patella tendon stretch reflex, okay, or called patella reflex, from the tapping of the patella with the reflex hammer. This draws patella momentarily down, simulating the muscle spindles, efferents, and Golgi tendon organs by altering the stretch and muscle length that provokes pre-programming reflex. So it gives you the reflex based upon that effect of the tapping. Okay, it's pretty interesting because they want to see whether or not you are functioning, your nerves are functioning properly relative to this. That's why they do that. Okay. They want to see if the muscle spindles, afferents, and Golgi tendon organs stretch and then reflex relative to that. And that has to deal with functioning in the proper functioning in the PNS. So here you have cross extensor reflex, where you step on a tack, pierces left foot, you step back. So what it does is it's called cross extensor reflex, what causes your right leg to extend. So your left foot, the tack pierces left foot, left leg goes back, then it extends the right leg, okay? And so the tack stimulates pain receptors in the skin. Receptors transmit message to the spinal cord via sensory nerve. Sensory neurons branch to each side of the cord to activate interneurons in the gray matter. Interneurons synapse with the motor neurons innervating both flexor and extensor muscles in each leg. Inhibition and stimulation of the appropriate leg flexor extensor muscles cause concurrent rapid extension of the uninjured limb inflection to remove the injured limb. Interneuron connections simultaneously activate neural pathways, transmit information to the appropriate sensory areas of the brain where the pain is felt. It's very interesting. So integration of sympathetic and parasympathetic nervous system. Most organs receive simultaneous parasympathetic. So most organs receive both the sympathetic and parasympathetic stimulation. Both maintain a constant degree of activation called neural tone. Depending on physiological need, one system becomes more active while the other becomes inhibited, allowing for finer level of control at the end organs. Now this here has to do with physiological need. Okay, do you need to elicit a sympathetic response? Well, I see something coming toward me, I need to hurry, and heart rate goes up, and pupils widen. Sympathetic response. Parasympathetic response is more of a rest and relax, where I do not need to elicit a quick response. Nerve supply to muscle. About 420,000 motor nerves exist, yet a single nerve usually supplies numerous individual muscle fibers. Ratio of muscle fibers to nerve generally relates to muscle's particular movement function. Basic role is that less complex movements have higher ratio of muscle fibers to motor nerves, while complex hand movements require more specialized, have much lower ratios. So, for instance, when you talk about muscle fibers to nerves, you have 10 to 1 for the eye, as opposed to the gastrocnemius, which is 1800 to 1. Okay, so the ratio, the number of muscle fibers per motor neuron, termed the innervation ratio, generally relates to a muscle's particular movement function. So the smaller the less complex movements, higher ratio. While complex eye and hand movements have a lower ratio. So lower ratio is like 10 to 1. You have the muscle fibers to nerve versus 1800 to 1 for the complex movement or high power movement of the gastrocnemius. gastrocnemius. So you have nerve supply to muscle continued. You have alpha motor neuron cell or cell body. You have dendrites, direction of propagation of action potential. This here is really just talks about how whole muscle contains numerous motor units, each with a single motor neuron and its complement of muscle fibers. The myelin sheath increases the speed of nerve impulses. It goes down, and at the motor end plate has the acetylcholine, causes the stimulation of whatever muscle that is. It's a really quick way to talk about it. You have at the end of those so at the end of the down here, which is what this is showing right here, this what you have here is you have this motor end plate, okay, terminal branches of the dendrites. And that's what this is here, going into a muscle sarcolemma or sarcoplasm of a muscle fiber. So this is going into a muscle fiber, okay? Neuromuscular junction, acetylcholine, action potential, action. Okay. We'll talk a little bit more about that. So basically, this is the microanatomy of neuromuscular junction, including details of presynaptic, postsynaptic contact, the area between the motor neuron, muscle fiber, innervates. Inset table shows a representative value for ionic concentrations across the motor neuron membrane. Synaptic vessels causing acetylcholine, and then that enables the ability for the muscle to twitch or move. So motor unit, skeletal muscle fibers and their corresponding innervative anterior motor neuron. Functional unit of movement. Whole muscle contains many motor units, each of which contains a single motor neuron and its composite muscle fibers. Motor unit anatomy consists of cell body, houses the control center. Axon extends from the cord and delivers impulse to the muscle fiber innervates. Dendrites receive impulse to the spinal cord connections and conduct them toward the cell body. Nerve cells conduct impulses in one direction only down the axon, away from the stimulation point. And all the motor units, muscle fibers, disperse over subregions of the muscle with other motor unit fibers. Myelin sheath encircles axon nerve fibers, keep the electrical current from leaving the bare axon while allowing high signal transmission. And that's what we showed the picture of, that myelin sheath. Schwann cells in the peripheral nervous system encase bare axon and spiral around it. Nodes of Ranvier account for higher transmission velocity. By the way, MS, myelin sheath, interferes with vision, sensation, body movements, the destruction of the myelin sheath, and it creates a peripheral neuropathy, which adversely affects neural pathways to the brain, spinal cord, and optic nerves, which is what MS does. That's the disease that causes those neurological functioning problems. Neuromuscular junction or motor end plate provides interface between the end of a myelinated motor neuron and muscle fiber. Functions to transmit nerve impulses to muscle fibers. Presynaptic terminals lie close to the sarcolemma. Synaptic cleft is the region where the neural impulse transmit occurs. Excitation occurs at the neuromuscular junction. Acetylcholine provides this chemical stimulus to change an electrical neural impulse into a chemical stimulus at the motor end plate. Facilitation. All right, so this is a motor neuron. A motor neuron generates an action potential when its microvoltage decreases sufficiently to reach its threshold for excitation, effectively disinhibiting. This inhibition fully activates muscle groups during maximal lifting. Enhanced neuromuscular activation accounts for considerable improvements in neuromuscular strength without concurrent increases in muscle size. Inhibition. Some presynaptic terminals generate inhibitory impulses by releasing chemicals that increase postsynaptic membrane permeability to potassium chloride ions. This increases membranes resting electric potential to create an inhibitory postsynaptic potential that makes a neuron more difficult to fire. And gamma aminobutyric acid and glycine exert inhibitory effects. Neural inhibition serves to protect functions and reduce input of unwanted stimuli to produce smooth, purposeful responses. Okay. So example, one can usually override or inhibit the reflex to pull the hand away when removing a splinter. Okay. So because you don't want to pull the hand away when you're removing the splinter because you're trying to remove it. All right. You have motor unit characteristics, twitch characteristics in response to single electrical impulse. Some units develop high twitch sensors, high twitch tensions, while others develop low twitch tensions, and others generate intermediate tension. So tension generating characteristics, different motor units, muscles that innervate or can develop different amounts of tension based on many factors, governed by three factors: all or none principle, graduation of force principle, and level of motor unit recruitment patterns. Okay, all or none principle. This is basically Harvard's Medical School Department of Anatomy and Physiology and Physical Training. This is where in 1871 Henry Pickering Bowditch, renowned American physiologist at Harvard, came up with this. Basically is that if a stimulus triggers an action potential in the motor neuron, all the accompanying muscle fibers must contract synchronously. A single motor unit cannot generate a strong or weak contraction. So there is no strong or weak. It either elicits a contraction or it doesn't. Once a neuron fires and the impulse reaches neuromuscular junction, the muscle cells always contract to the fullest extent. Gradation of force principle. Force of muscle action varies from slight to maximal by two mechanisms: increasing number of motor units recruited and increasing frequency of motor unit discharge. Activation of all motor units in a muscle generates considerable force when compared with activating only a few units. So total tension also increases if repetitive stimuli reach a muscle before it relaxes. These range from the delicate touch of an eye surgeon repairing a retinal blood vessel to the maximal effort in throwing a baseball fast. And there's, on page 336, it talks about that with the tremendous complexity of neural patterns involving millions of bits of information required to throw the baseball in a coordinated fashion. Okay, so process of adding motor units to increase muscle force. You have size principle. Motor neurons with progressively larger axons become recruited as muscle force increases. Selective recruitment and firing pattern of the fast twitch, slow twitch motor units control movement, provide the mechanism to produce the desired coordinated response. So all of a muscle's motor units don't fire simultaneously because, you have to, when you're lifting a barbell, for example, you're going to move the limb at a particular speed under a set rate of tension. You're not going to do fire all the motor units and do it super quickly. Like when you lift a pencil, you're going to generate just enough force to lift the pencil regardless of how fast or slow the arm moves. Okay. When lifting heaviest weights, all of the available motor units require activation. Okay, so weightlifters you basically have the synchronous firing of motor units, and then you have people like doing jogging, cycling, cross-country skiing. They have more of an asynchronous pattern. So neuromuscular fatigability. This represents a decline in muscle tension or force capacity with repeated stimulation per unit time. Resistance to fatigue is defined as the maintaining muscle tension with repeated stimulation represents another important quality to distinguish differences in motor units. factors that decrease the force generating capacity. You have exercise-induced alterations in levels of CNS neurotransmitters, serotonin, 5-hydroxy, tryptamine, dopamine, and acetylcholine. Reduced glycogen content, that obviously causes neuromuscular fatigability in active muscle fibers during prolonged exercises because you run out of that, and then how do you have any type of ATP source? So you have, and that, which, that's what carbohydrate is. So you have an increased level of blood and muscle lactate, which then causes fatigability because pain if you can't get rid of that muscle lactate, then fatigue at the neuromuscular junction. Proprioceptors. These are receptors to stretch, tension, and pressure. Specialized sensors that they in the muscles and the joints and tendons. They relay critical information about muscular dynamics, limb position, and kinesthesia, human movement, and proprioception to conscious and subconscious portions of the CNS. Allow continual monitoring of the progress of any movement or sequence of movements and serve as the basis for modifying subsequent motor actions. Proprioception, this talks on about the so you have, in regards to proprioceptors, you have these peripheral nerve motor and certain sensory nerve fibers, and it's dissecting it, so it shows that in sensory neurons you have the muscle spindle, and then you also have Golgi tendon, and what happens is that the muscle spindles are responsible for stretch. They provide a mechanosensory information about changes in muscle fiber and tension, and then you have the Golgi tendon organ that deals with the tension of the muscle. We'll talk about that shortly. Okay, we'll just go into that a little bit. So we have muscle spindles. These provide mechanosensory information about changes in muscle fiber tension and length. They respond to muscle stretch through reflex action by initiating a stronger muscle action to counteract the stretch. So this is like if you are given a dumbbell and it's weighted, and then your arm starts to drop, and you can, first it starts to go down, but what catches it is that stretch. Okay, the reflex action counteracts that stretch. That's a good example. More spindles exist in muscle than routinely perform. More spindles exist in muscles that routinely perform complex movements. Okay, so they have more spindles. Stretch reflex. The stretch reflex consists of three main components: muscle spindle that responds to stretch, afferent nerve fiber that carries sensory impulse from the spindle to the spinal cord, efferent spinal cord, efferent spinal cord motor neuron activates stretched muscle fibers. So is exactly what I said. You have someone puts a dumbbell of 20 pounds in your hand. So your arm starts to drop a little bit. You have the afferent nerve that carries the impulse from the spindle to the spinal cord. Efferent then gives you that stretch. It activates the stretched muscle fibers. Okay. And that's just talking about this here. You have what goes on here. You have the afferent that goes from the muscle fiber to the spinal cord, and then you have the inhibitory interneuron that then causes the alpha motor neuron to then cause that stretch reflex. Golgi tendon organs connect a series of extrafusal fibers and also located in ligaments and joints, primarily detect differences in muscle tension rather than length. When activated by excessive muscle tension or stretch, the Golgi receptors immediately transmit signal to cause a reflex inhibition of the muscles they supply. This protects the muscle and connective tissue harness from injury by sudden excessive load or stretch. So if you get that tension that's placed on the muscle after that stretch, it inhibits that tension. What it does is it transmit signals to cause that reflex inhibition of the muscles they supply. So It protects muscles from that stretch by giving tension to the muscle. Okay. So, and this is what this Golgi tendon. This is another. This here actually talks more about the bone, tendon, muscle belly, how a muscle looks, and then the interior of what the sarcolemma is. So we can come back to that. But Pacinian corpuscles is the next thing that's in the book. Okay, and they respond to pressure, quick movement, detect change. Ellipsoidal bodies located close to the Golgi tendon organs and embedded in a single unmyelinated nerve fiber. They're found in subcutaneous tissue on the nerves and the soles of the feet, palms of the hand, mucous membranes, and male and female genital organs, and in close proximity to the other nerve joints. These detect changes in movement or pressure rather than magnitude of movement or amount of applied pressure. Okay, so they respond quick to movement, and deep pressure. Skeletal muscle structure, and that's what the picture was previous. That this is epimysium, perimysium, endomysium, sarcolemma, sarcoplasm, sarcoplasmic reticulum, and that's what this is. So it just goes from large to small. You have muscle, bundle muscle fibers, single muscle fiber, to the myofibrils. You have the sarcoplasmic reticulum, that's calcium, and we'll talk a little bit about this. And you have to read through this in your book because it's a good bit of information. It's on the page. It's levels organization, but basically you have the epimysium, which is a fine layer of connective tissue wraps each muscle fiber, separates from neighboring fibers. Another layer of connective tissue is perimysium. Then you have the fasciculus, and then you have the epimysium, surrounds the entire muscle with fascia or fibrous connective tissue. So it's like a cascade. Okay. Skeletal muscle capillarization. So you have capillaries is for microcirculation. Expedites removal of heat and metabolic byproducts from active tissues. Provide large surface area to exchange not only metabolic generated heat, but fluids, electrolytes, gases, macromolecules. Total number of capillaries for muscle averages about 40% higher in endurance-trained athletes than untrained counterparts, which isn't surprising because they are using the oxidative. They are doing aerobic activity, okay, where they need capillaries. So that makes sense. And then positive association exists between VO2 max and average number of muscle capillaries. Skeletal muscle ultrastructure, you have the myofibrils, myofilaments, and actin and myosin. And that's what this is here. So you have myosin, which is the red, actin, which is the blue. These are the small, these are the large. And what happens is, this is the muscle fiber, and whenever these, and we'll talk about it, these myosin heads attach with these troponins and they pull on them, it's like a twisting effect, which cause these to overlap in the sliding filament theory. So sarcomere comprise the functional unit of muscle cell. Actin, myosin filaments provide the mechanical mechanism for muscle. Position of the sarcomere is that thin actin and thicker myosin proteins overlap the two filaments. The center of the A band contains the H zone, a region of lower optical density because of the absence of actin filaments in the region. The M line bisects the central portion of the H zone and delineates the sarcomere center, and the M line contains the protein structures that support arrangement of myosin filaments. So if you just look back here, that's what this is talking about. Okay, the Z line. Okay, you have the I band, you have the M band is in the middle, you have the A band, okay, that does not include the M band, but if the A band on both sides of the M band is where the actin and myosin are on top, like lined up. Okay, this is right here. So you have the A band, like I was showing you, the H zone, okay, and the Z line. Whenever muscles contract, you see this here. Oh, look at this. You have that actin, myosin, sliding filament. And this is like a cutout of what this is. So it's a cross section of myofibrils, which is representation electron micrograph through a cross sectional myofibril single muscle fiber. Note the hexagonal orientation of the smaller actin and larger myosin filaments, including the cross bridges that extend from thin to thick filaments. Okay, and this is really what this would see is this is like the I band here. These are just these are when they're not these here are when they're outside of each other. So it's like this would be a slice right here. So like this right here would be a slice right here of the Z line. Okay. You have here, you have this if I took a slice right here, so if I took a slice right here, that would be this right here. Okay. So, and then right here, these right here would be like something like really what you see is you see these here. This is where these are not they're there, but they're not overlap. They're not having that cross bridge. They're not cross bridging, right in here. The cross bridge formation isn't there. So sliding filament theory, muscle fibers shorten or lengthen because the thick and thin microfilaments glide past each other without filaments themselves changing length. Myosin cross bridges were cyclically attach, rotate, and detach from the actin filaments with the energy from ATP hydrolysis. So ATP hydrolysis, if there's ATP, it causes this attachment, rotation, detachment from the actin filaments, providing molecular motor to drive fiber shortening. Let's see. Okay, sliding filament theory. So these slide on top of each other, and this troponin head basically attaches to the myosin head from the myosin filament, which is red, attaches to the actin, and then the cross bridge formation that takes place in it. It attaches to troponin and then pulls it almost and slides past each other. That's really what takes place. So the cross bridges, the globular head of the myosin cross bridge provides a mechanical power stroke for actin myosin filaments glide past each other. During muscle activation, each cross bridge undergoes repeated but independent cycles of attachment to detachment to actin. A single cross bridge moves only a short distance, so cross bridges must attach, produce movement, detach thousands of times to shorten the sarcomere. And that's why you have all these different myosin heads, not just one, but a bunch. So this is what you have. You have all these myosin head, ATP here. Guess what? This here, myosin head, cross bridge attaches to troponin complex. Then the trypomyosin, it will slide down into the trypomyosin groove and then create that power stroke. Relaxed, and then this is shortened. And shortened would be like when there's a bicep curl, okay? And the calcium there is what's needed to make sure that takes place. Cross bridge, continue. This is just showing a muscle biopsy of human vastus lateralis, which is on your the lateral aspect of your thigh, your quads. It's just talking about the different muscle fiber types. Type 1 is a slow twitch. Type 2A is slow twitch but somewhat oxidative. And then you have X, and then C is a rare. But this is actually the fastest glycolytic. Why these are like this is the pH. You have the fibers are stained for myosin ATPase activity, which then that's why you're seeing there's the whiteness from you're seeing the acidity in C. Well, these things in the book actually are showing that the myosin. So if you look at this one here, okay, example, myosin ATPase stained white for acidity, pH of 4.3. See the acidity is there. Myosin ATPase, acidity, and that's why you see these fiber types. Okay, because that way you know that's the myosin ATPase, where there's an acidic environment, and that's what it's staining it for, is the fast twitch muscle fibers. So here you have, this talks about fiber type 1, type 2A, type 2X, type 2B. So you have type 1, slow contraction time. Resistance to fatigue is high, so you can go for a long time. It's just the firing rate is slow. That's why it's, type 1 slow twitch. And then you have high capillary density. Makes sense. It's oxidative. Type A fibers. Type A, you have moderately fast, long-term anaerobic. Okay, this might be something like a 400-meter sprint. It's firing, but not as fast as it could with like the X right here, which is a fast twitch, short-term anaerobic, less than 5 minutes. And then type 2B, which is very fast. Okay, short-term anaerobic, less than 1 minute. Okay, so capillary density really low. It's not oxidative. It's dealing with obviously like less than 1 minute. I mean, you're dealing with glycolytic system, which is anaerobic. Fast twitch muscle fiber characteristics. You have rapid transmit of action potentials, high activity of myosin ATPase, rapid rate of calcium release and uptake by sarcoplasmic reticulum, generate rapid cross-bridge turnover. That's why it's labeled as fast. One of the things too about and I have to mention this, that because it's just kind of interesting. And you need ATP, by the way, to have this attachment and detachment of these cross bridges to the troponin. Muscles actually, on page 354, it talks about rigor mortis. And rigor mortis is when muscles become stiff and rigid 3 hours after death. Well, that's because ATP no longer functions in the sarcoplasmic reticulum's membrane to pump calcium ions into the terminal cisternae. So, because when calcium diffuses from higher concentration to lower, it binds to troponin, allowing cross bridges, actin, myosin. So, without the ATP, myosin cross bridges and the actin remain attached. That's what happens with rigor mortis after death. The ATP is not there to release these cross bridges. That's why people get stiff. Okay, it's interesting. It's a physiological thing that takes place. All right, back to this. So fast switch subdivisions. Talked a little bit about this. We have type 2A fiber, exhibits fast shortening speed, moderately well-developed capacity for energy transfer, aerobic, anaerobic sources, represents fast oxidative glycolytic. Then you have type 2X. Possesses the greatest anaerobic potential and most rapid shortening velocity, represents fast glycolytic fiber. And then C, are normally rare, undifferentiated, may contribute to reinnervation and motor unit transformation. Slow twitch muscle fibers generate energy for ATP resynthesis predominantly by aerobic energy transfer, possess low activity level of myosin ATPase, slow speed of contraction, glycolytic capacity less well developed than fast twitch counterparts, resist fatigue because they're oxidative and they're slow twitch. I mean, it's, that's why but they don't elicit a much power, and the power so the power is prolonged in aerobic activity. Slow oxidative fibers. Muscle fiber type distribution differences. So muscle fiber type distribution differences, individuals, genetic code is genetic code is a primary determinant of a person's predominant fiber type. You can train this. I was told in the motor development book about 2 to 9% of these can be trained right after birth. They can be, and you can, specific exercises train and improves the metabolic capacity for each fiber type. However, once a fiber type is like fast twitch, they can fluctuate between, let's say, if you're doing repetitive explosive activity, they can be trained to be like type 2X. If you're doing 400-meter sprints, then you're going to have a higher percentage going towards type 2A because they're still they're going toward toward the oxidative type 2 muscle fiber type. However, the slow twitch type 1 is the where I'm going with it is within the muscle fiber type it can be trained, but you can't have a percentage of type 1, let's say slow twitch, and then that's changing over to type 2. No, you can only change within the fiber type. They can't be changed between through training. Okay, so that's interesting, right? But the fiber type can also get larger relative to the training you're doing. So if you're doing a lot of distance running, your type 1 fiber type can actually grow diameter wise. It's the same thing with type 2. They can change percentage from type 2A to type 2X, let's say, if you're doing more short-term sprinting, 40-yard dash, 20-yard dash, things like that. Explosive plyometrics. And those can widen as well. However, like the diameter, but they can't change between type 2 and type 1, the percentage.